Hello students, welcome to my channel. We are on video number 4 for chapter number 11. We are discussing few important questions from that chapter. Question number 1 is asking Agrobacterium tumefaciens is called as the natural genetic engineer. Now why is it known as natural genetic engineer? Because it does gene transfer without any human effort. So whenever we are dealing with any plant cell, then generally this is being used. The DNA fragments in an agarose gel at the same position signified that the two DNA fragments have same molecular weight. So this question may come indirectly as that if you have observed DNA fragments in an agarose gel on the same position, what will you interpret? So you need to explain here the principle of gel electrophoresis, that it works on the phenomenon of molecular weight, that the DNA fragments will get separated based upon the molecular weight. The one who will have smaller molecular weight will be found near the anode region, and the one who will have higher base pairs or more molecular weight, they will be found near cathode. So this is important. Restriction endonucleases are used in the genetic engineering to make the recombinant molecules of DNA. Here the GOI is written, which means gene of interest. Okay, so gene of interest is able to multiply in the bacterial cell because of the ori site. Remember, whenever we are choosing any vector, we need to be assured that it should have a ori site, because the gene of interest will be ligated onto it. As soon as the ori site will open for the DNA replication, the gene of interest will also be replicated. Molecular scissors are restriction enzymes. If it is asked what are molecular glues, then these are the glues. Phosphatases are used to stop the self-ligation of the gene of interest in the plasmid DNA, and polymerase is one of the enzymes which is used in the elongation process of DNA replication. Okay, each enzyme with its role should be understood to you, because shortly you will have your NEET examination. Agarose gel is used in electrophoresis because it is inert. Inert means inactive and it does not react with DNA. Because of the same purpose we use it. ROP genes in PBR322 codes for ROP was one of the segment that was close to ori site. So these helps in producing the proteins that helps in the replication of the plasmid. What type of cut ends are formed when both the strands of DNA are cleaved at exactly the same nucleotide position. So if they are cutting at the same position, suppose we have CCCGGG, then if it is cutting like that, then these ends are equal, and such ends are known as blunt ends. And SMA1 is an example of the restriction endonuclease that produces such ends. Selectable markers are the antibiotic resistant genes, which are quite helpful in the transformation process because they will help us in separating the recombinants from the non-recombinants, transformants from the non-transformants. You can give an example of PBR322, which is a widely used vector. It has two selectable markers, ampicillin resistant gene and tetracycline resistant gene. Okay. Give an example of microorganisms that have transforming ability. So Agrobacterium tumefaciens is the answer to it. What is a vector? Vector is a DNA molecule that has the capacity to take another DNA molecule, and while cloning vector has to be chosen, then one should always remember that it should have a definite ori site. Because of the ori site only the gene of interest gets expressed. Then they are asking which cloning vector was discovered first. So here you'll be writing PBR322. Here P refers to plasmid. BR are the names of discoverers. Names of discoverers. B is Bolivar. R is Rodriguez. Rodriguez. and 322 is the molecular weight. Okay, molecular weight in kilodaltons. In kilodaltons. When this question will come, it would be expected. If you could do this, it would be great and it will give you full on full. What is recombinant DNA? The DNA from two different organisms, when it is ligated together, it is known as recombinant DNA. Whenever we mix the gene of interest with the plasmid DNA, it forms the recombinant DNA. For creating recombinant vector molecule, can two different restriction enzymes be used for cutting vector in a source DNA? Not at all, because it will produce different sticky ends and the process would be a fail. So you can explain by taking the example of eco R1, that it acts upon GART. TC is the palindromic sequence which is being recognized, and this is the recognition site, right? So you can explain how sticky ends are being formed. So in the gene of interest and in the plasmid DNA, same sticky ends are formed, so the probability of unison is quite strong, and this will help in forming the recombinant DNA. So if we are going to use different restriction enzymes, then recombinant DNA will not be formed. Right? Question number 13 says a newspaper has reported that an American company has patented turmeric. Indian government is fighting against this patent. What will you call this act of American company? So it is truly an act of biopiracy. Here you have already been provided with the literature at the back about rice, how many strains India do have, and India is the producer of basmati rice, for which India is being known. So you can say about one of the case being described in the book itself. Write the full form of PCR. Is polymerase chain reaction. What are the three basic steps? So it is denaturation, it is annealing, it is extension. Again, I would like to request everyone that please do mention temperatures here, which are very, very useful, and what all happens in this process. So in denaturation, the double helical DNA strands, they move apart from each other, and this marks the end of the denaturation. The instrument which is used for PCR process is known as a thermocycler. So in a thermocycler, all the contents are being fed. In denaturation, the two strands move apart from each other because of the high temperature. In annealing, the temperature is 50 degrees Celsius. Two primers are required, one the forward primer, another the reverse primer, right? Or you can simply name primer 1 or primer 2, right? For this, I'll be sharing a link in the I button. You can go through the process. For the elongation process, we would be requiring Taq polymerase, deoxynucleotide triphosphates, which will be picked up by the DNA polymerase, and they will help in elongation. RAS process is describe briefly the following origin of replication. So origin of replication is a site which helps in opening the DNA and forming the replication fork. So this is the ORI site which helps in opening the DNA, and it helps in synthesizing the daughter strands. On one strand the synthesis is continuous, on another strand it is discontinuous. The replication of DNA is semi-conservative in nature because one of the strand in the daughter DNA is that of the parent and other one is the new one. So whenever we are choosing a vector, it should have its own ORI site because this will help in accommodating the source DNA, and as the replication of the vector DNA happens, the replication of the source DNA will also happen. I hope all the questions are being understood. Please like, share, subscribe to my channel, that is Online Biology Tutorials. I have one more video on chapter number 11. Stay connected. Thank you.